if we could communicate with life, communicate with the cells that make up our bodies at their level? Would we be able to rewrite diseases, reprogram the way cells behave? Would we be able to create little nanomachines that can work together with our own proteins? I want to introduce you to two different examples that I've been involved in where we've been looking at DNA origami as a potential medicine. I work in the field of nanotechnology, and this encompasses all medicine and technology, but at the scale of a billionth of a meter. To put this into perspective, a single sheet of paper or a human hair are about 100,000 nanometers wide. Here it is, picture of my 100 nanometer nanoparticle. I can't see it either. And if you could, you'd have eyes like an electron microscope. But if we magnified this 10 million times, we would be able to see it, but it would be the size of a Cheerio. So after 10 million times magnification, we're talking about something which is this big. You are made of trillions of cells: skin cells, kidney cells, lung cells, heart cells, all working together in concert. Within each of these cells is a copy of your DNA. Now, just like a computer reads ones and zeros to create complex programs, your cells read your DNA to create little nanomachines and workers that we call proteins. And these enable your cells to function. But sometimes life gets it wrong. And it also happens that if you change the sequence, or if you change the way your cell is reading that sequence, is how we get many diseases. Over 5,000 diseases occur because of one single letter wrong. And this can lead to things such as various cancers, Alzheimer's, heart disease, cystic fibrosis. DNA origami is where we take synthetic, arbitrary DNA sequences and we fold them into nearly any 2- or 3-dimensional shape that we want. We do this by taking one large piece of DNA, and then we design small pieces of DNA which interact with different regions of that larger piece. So when they bind and they form their complementary pairs, it actually pulls it together to create a fold. So this is one short sequence and one longer piece of DNA. But now just imagine what happens when we have hundreds of those smaller DNA sequences, all pulling that larger DNA in different directions. Now we can start to get really complicated designs. Now we can start to get molecular rotors and machines and even DNA walkers that can walk along surfaces. And these designs are all written in the sequence of those short strands. So they're all pre-programmed. Which means, I'm sorry to break it to you, but I don't go to the lab and sit there underneath a microscope looking and trying with ultrafine tweezers to manipulate a piece of DNA. Because the reality is that you mix these in a tube and it self-assembles. It forms itself. In the first example, we created a shell. This shell is made of 10 smaller DNA origami triangles which fit together in a particular way, like Lego bricks or puzzle pieces. Again, by self-assembly, there's only one way this can form. This is a reconstruction from real microscope images that we've taken. And what you see running along here, these little tubes, those are DNA helices. But why make a shell? We did it to trap viruses. What you can see is a virus sitting nicely nestled in the center of our shell. And what happens when the virus sits there is our shell actually physically blocks the virus from being able to interact and infect cells. So effectively, we've blocked and stopped the virus life cycle. But I wanted to take it one step further. DNA encodes for biological messages, for instructions. We wanted to know if we took a DNA message and we folded it into an origami particle, gave it to a cell, would that cell be able to unfold it, read it, and use it? So here, I took the DNA blueprint for a green fluorescent protein. That is, the sequence of DNA, which when the cell reads it, can create a protein which fluoresces green. Now we took that sequence and we bundled it up, we folded it into this nice origami particle that you can see here. It's beautiful, right? These stripes, again, these are the DNA double helices that you're seeing packed together. When this origami particle goes into the cell, if the cell is able to read and use that message, the cell should turn green. And that's exactly what we saw. Here we have individual cells. Each of these little squiggly shapes is an individual cell reading that DNA origami message and making those green proteins. Not only do we have immense control over the size and the shape of these particles, but we can actually program useful biological messages into them. More than that, we can piece together multiple messages, like a puzzle piece, like a Lego brick that I showed you earlier with the shell. But why is it important? Why would we want to put together multiple messages? It's the unfortunate reality that when you're looking at treating complex diseases, such as cancer, one of the things that makes it so complex is it is not a single disease, even within one person. Those cancer cells are present all at different stages with different characteristics, different behaviors. Some might want to stay tightly packed within a tumor, while others will move and want to move around the body. And sometimes it can be problematic if we try and treat them in the same way. So, looking into the future, what if we could deliver a DNA message, like a therapeutic gene, into a cell to rewrite it, reprogram the way that cell is behaving? But not only that, what if we could change the message depending on which cell it entered? I'm sure you all remember these. At school, you go through a sequence, your color, a number, a name, and then your friend would unfold the origami and reveal your future. And then your friend would do it next to you, and another part of the origami would unfold, and their future would be read. What if our therapies could be similar? What if we could unfold and read and deliver a message that only that cell needs? What if we took a DNA particle with multiple messages inside it, and then when it got into one cell, it was able to unfold one message and deliver it? But that same particle, if it got into a different cell, would recognize it needs to unfold this second part and deliver this second message to that cell. This is what I think the future of our medicine could look like. Myself, I see the huge potential here we could have to change the way that we treat diseases and maybe even cure them. Thank you.